The topic of today's lecture is pharmacodynamics, and pharmacodynamics uh, is what drugs does to the body. And most of the drugs act on the body through the receptors. So the receptors will bind with the drugs, and then that will initiate a biological response. So depending on this biological response, there may be a different action, and that is the most common way of the drug action. The And usually when the receptor is free and unoccupied, it will not lead to any biological response. And when the drug will bind to the receptor, to the special site of the receptor that is binding site, and then receptor is activated, and that leads to the biological response. And the result of this biological response depends on the cell and on the tissue, and sometimes the receptor, depending on the type of the receptor, there may be a different intracellular effect. So if the receptor is the ion channel, then the activation of the receptor by the drug here. So this is a drug that will bind the binding site of the receptor that is connected with the ion channel, and then that enables that enables the ions pass through the channel. There are four main receptor families, and all the receptors are proteins that will transform the extracellular signal to the intracellular response. And four families include line-mediated ion channels, G-protein coupled receptors, enzyme link receptors, and intracellular receptors. And one moment please. So and the receptor uh, receptor activation uh, receptor activation leads to the some intracellular effect. The intracellular effect depends on the type of the receptor, and the the ion channels uh, usually contains a ligand binding site that is shown here, and that this site regulates the uh, state of the receptor. It can be open or closed, and usually the channel is closed if the receptor is not activated by the agonist. And depending on the ion conducted through these channels, these receptors may lead to different results. And for the sodium and potassium, that's usually attraction or relaxation. So also for the chlorine, it can be the excitation of the neuron. So for example, GABA receptors on the central nervous system will promote uh, the neuron excitation decrease, and these an example of the allyl gated ion channels is a cholinergic receptor, and cholinergic receptor is activated by the nicotine, and that results in the sodium influx and potassium. So sodium that is outside will go inside, and then potassium that is inside will go outside. And that leads to the activation of the neuron, and neuron is usually connected with the skeletal muscle, and then the muscle will contract. So in the central nervous system, it's vice versa. The gamma aminobutyric acid, or GABA receptor, increases fluorine influx, and that because fluorine is negatively charged, it will enter the cell and make it even more negatively charged, and that will hyperpolarize the neuron, and neuron will not be activated frequently. Voltage-gated ion channels may also possess ligand binding site that can regulate channel function. For example, local anesthetics bind to the voltage-gated sodium channel, inhibiting sodium influx and decreasing neuronal conduction. So the second type of the receptor is the transmembrane G-protein coupled receptor, and the extracellular domain of this receptor contains the line binding area, and the intracellular domain has a G protein, and there may be different kind of G proteins. Some of them are inhibiting, some of them are stimulating, but they are composed of the three protein subunits alpha, beta, and GABA, and alpha subunits binds to the guanosine triphosphate, and the beta and gamma subunits bind in the cell membrane. Binding of an agonist in the receptor increases guanosine triphosphate binding to the alpha subunit and causing dissociation of the alpha GTP complex from the B gamma complex. These two complexes then interact with other cellular effectors, usually an enzyme, a protein, or an ion channel that are responsible for further actions with the cell. So these receptors are slower than the ion channels because ion channels are fast, like several seconds. These are several minutes, and so these receptors can have second messages that activate them, and they activate other functions in the cell. So and the most common activated effector is an adenylate cyclase, that is an enzyme which produces the second messenger cyclic adenosine monophosphate, and also the phospholipase C that generates the two other second messages, inositol 3-phosphate and deacylglycerol. These two can activate different kinases within the cell, leading to some physiological effects. Inositol 3-phosphate regulates intracellular free calcium concentration as well as some protein kinase activity, and calcium concentration is usually the increase usually leads to the increase in the muscle contraction, and decrease is usually leads to the relaxation. So the third type is enzyme-linked receptors. This family of receptors consists of a protein that may form dimers or multi-subunit complexes. When activated, these receptors undergo conformational changes, resulting in increased cytosolic enzyme activity. Depending on the structure and function, this response lasts 1 hour or maybe several hours. So the most common enzyme-linked receptor possesses tyrosine kinase activity as part of their structure. The activated receptor phosphorylates tyrosine residues on itself and then other specific proteins. Phosphorylation can substantially modify the structure of the target protein, thereby acting as a molecular switch. So the common example is insulin that binds to two of its receptor subunits. Their intrinsic tyrosine kinase activity causes autophosphorylation of the receptor itself. In turn, the phosphorylated receptor phosphorylates other peptides or proteins that subsequently activate other important cellular cells. This cascade of activations results in a multiplication of the initial signal, which like that with the G protein coupled receptor. So these two types of receptors, G protein coupled and enzyme linked receptors, have many common features. So, and the last type is intracellular receptors or steroid receptors. That is the main family for the steroid receptors. These receptors are different from the previous ones because they are intracellular and uh, they are intracellular and the to activate the receptor, the drug must diffuse into the cell to interact with the receptor. In order to move across the target cell membrane, the ligand must have sufficient lipid solubility, and the primary targets of these ligand receptor complexes are transcription factors in the cell nucleus. Binding of the ligand with its receptor generally activates the receptor via dissociation from a variety of binding proteins. The activated ligand receptor complex then translocates to the nucleus, where it often dimerizes before binding to transcription factors that regulate gene expression. The activation of these factors causes the transcription of DNA into RNA and translation of RNA into an array of proteins. The time course of activation and response of these receptors is on the order of hours to days and can be as long as weeks. For example, steroid hormones exert their action on target cells via intracellular receptors. Other targets of intracellular ligands are structural proteins, enzymes, RNA, and ribosomes. For example, tubulin is the target of antineoplastic agents such as pac-B-Paxel. The enzyme dihydrofolate reductase is the target of antimicrobials such as trimethoprim, and the S50 subunit of the bacterial ribosome is the target of microbiotic antibiotics such as erythromycin. So then the uh, the uh, the repeated administration uh, of the drug can lead to the decrease in the activity, and that is called a desensitization. So desensitization is a decrease of effect uh, over a short time period and following the repeated administration. So this is the response of the for the drug from the first administration, and then this is repeated administration, and then the response is slightly less. So that is because of the desensitization of the receptors. So, but if we wait for a long time and then administer the drug again, then the response will recover. So, but that needs some time for the rest. And also if the receptor is if continuous administration can lead to the down-regulation of the receptor, and down-regulation is the decrease in the number of the receptors. So this phenomenon that is called tachyphylaxis is due to the phosphorylation or other event that modifies the receptor on the cell surface and makes it unresponsive to the drug. In addition, receptors may be down-regulated such that they are internalized and sequestrated within the cell and become unavailable for further drug interaction. These receptors may be recycled to the cell surface, restoring sensitivity, or may be further processed and degraded, decreasing the total number of receptors available, like what happens during the Parkinson's disease. Some receptors, particularly ion channels, require some time following stimulation before they can be activated again. During this recovery phase, unresponsive receptors are said to be refractory. Similarly, repeated exposure of a receptor to an antagonist may result in upregulation of receptors, in which receptors reserves are inserted into the membrane, increasing the total number of receptors available. Upregulation of receptors can make the cells more sensitive to agonists and or more resistant to the effect of the antagonist. So the agonist drugs mimic the action of the original endogenous agonist of the receptor. For example, isoproterenol mimics norepinephrine on beta receptors of the heart. The magnitude of the drug effect depends on the drug concentration. So the drug concentration is on the x-axis and the effect is on the y-axis, and this is determined by the dose of drug administered and by the drug's pharmacokinetic profile, such as rate of absorption, distribution, metabolism, and elimination. So we discussed all of these 4 stages last week. As the concentration of the drug increases, so towards the right, so if we increase the concentration, its pharmacologic effect is also increasing, so it goes up, and until all the receptors are occupied, and at this point that is the maximum effect, and further increase will not result in the increase of the effect. So the further increase of the concentration will not result in the further increase of the effect. So this will be long, so continuously. The plotting the magnitude of response against increasing dose of a drug produces a great dose response curve. So this is that kind of curve, and usually the shape of this curve is hyperbola. So it is hyperbolic function. This curve can be described as a regular hyperbola, which is a familiar curve in biology because it can be applied to diverse biological events such as enzymatic activity and responses of pharmacologic agents. So, using this cube, two important functions can be detected for the drug: potency and efficacy. Potency is a measure of the amount of drug necessary to produce an effect of a given magnitude. The concentration of the drug producing the half of the maximal effect. So the maximum effect is 100%. Then we make the 50% from that, and then make a straight line that is parallel to the x-axis, and then at the point this line will cross the cube, then we will make a perpendicular line to the x-axis, and this is the EC50. So this constant will determine the potency of the drug, and the lower is this EC50, the more potent is the drug. So EC50 for drugs A and drug B indicate that drug A is more potent. So the red line, drug A is more potent than drug B, and that is orange, because the less amount of drug A is needed when compared to drug B to obtain the 50% effect. So this is lower than this. The therapeutic preparations of drugs reflect their potency. For example, the candesartan and irbesartan are angiotensin receptor blockers that are used to treat hypertension. The therapeutic dose range for candesartan is up to 32 mg as compared to 300 mg for irbesartan. Therefore, candesartan is more potent than is irbesartan. Since the range of drug concentrations usually spans several orders of magnitude, semi-logarithmic plots are used so that the complete range of doses can be graphed. As shown in the right figure, the cubes become sigmoidal, and this can be interpreted easier. So the cube is S-like, and EC50 can be detected more precisely using these cubes. And the second property is efficacy, and efficacy is the magnitude of response of a drug causes when it interacts with the receptor. Efficacy is dependent on the number of drug receptor complexes formed and the intrinsic activity of the drug. And the maximal efficacy of a drug assumes that all receptors are occupied. So because all the receptors are occupied, no increase in response is observed if a high concentration of drug is obtained. Therefore, the maximal response differs between full and partial agonists even when 100% of the receptors are occupied by the drug. Similarly, even though an antagonist occupies 100% of the receptor site, no receptor activation results, and maximum effect is 0. Efficacy is a more clinically useful characteristic that is drug potency because the drug with greater efficacy is more therapeutically beneficial than is one that is more potent. So if we look at the drug A, that it is more potent than drug B, but they have the same efficacy, and drug C has lower potency and has lower efficacy than drug A and drug B. So the effect of drug concentration depends on the receptor binding. The quantitative relationship between drug concentration and receptor occupancy applies the law of mass action to the kinetics of the binding of drug and receptor molecules. So if we suppose that the binding of one drug molecule does not alter the binding of subsequent molecules, and applying the law of mass action, we can mathematically express the relationship between the percentage of bound receptors and the drug concentration. So if we plot this function with the graph, then we will get this hyperbola. The mathematical model that describes the drug concentration and receptor binding can be applied to dose and response, providing the following assumptions. The magnitude of the response is proportional to the amount of receptors bound or occupied, and the second, the maximum effect occurs when all receptors are bound. The agonist binds to a receptor and produces a biological response based on the concentration of the agonist and the fraction of activated receptors. The intrinsic activity of a drug determines its ability to fully or partially activate the receptors. And drugs are usually one of the groups, like full agonist, partial agonist, or inverse agonist. Full agonist is a drug that binds to a receptor and produces a maximal biologic response that mimics the response to the endogenous ligand. It is a full agonist. Full agonists bind to receptors, stabilizing the receptor and its active state, and are said to have an intrinsic activity of the receptor. All full agonists for receptor population should produce the same maximum activity, so this is 100% of maximal activity. For example, phenylephrine is a full agonist at alpha-1 subtype receptors because it produces the same effect as does the endogenous ligand, and norepinephrine. Upon binding to alpha-1 receptors on vascular smooth muscles, phenylephrine stabilizes the receptor in its active state. This leads to the mobilization of intracellular calcium causing interaction of actin and myosin filaments and shortening of the muscle cells. The diameter of the arteriole decreases, causing an increase in resistance to blood flow through the vessel and an increase in blood pressure. As this brief description illustrates, an agonist may have many measurable effects, including actions on intracellular molecules, cells, tissues, and intact organisms. All of these actions are attributable to interaction of the drug with the receptor. For full agonists, the dose-response curves for receptor binding and each of the biological responses should be comparable. Partial agonists have intrinsic activities greater than 0 but less than 1. So this is a partial agonist and it has about 60% effect. Even if all the receptors are occupied, partial agonists cannot produce the same maximal effect as a full agonist. However, partial agonists may have an affinity that is greater than, less than, or equivalent to that of a full agonist. When a receptor is exposed to both a partial agonist and a full agonist, the partial agonist may act as an antagonist of the full agonist. Consider what would happen to the maximal effect of a receptor saturated with an agonist in the presence of increasing concentrations of partial agonists. As the number of receptors occupied by the partial agonist increases, the maximum effect will decrease until it reached the maximum effect of the partial agonist, so maximum will be at the 60%. This potential of partial agonists to act as both as agonists and antagonists may be therapeutically utilized. For example, aripiprazole, an atypical antipsychotic, is a partial agonist at selected dopamine receptors. Dopaminergic pathways that are overactive tend to be inhibited by aripiprazole, whereas pathways that are underactive are stimulated. Thus might explain the ability of the drug to improve symptoms of schizophrenia with a small risk of causing extrapyramidal adverse effects. But we will get to that later next semester. So, and the last one is inverse agonists. Inverse agonists are unbound receptors are inactive and require interaction with an agonist to assume an active conformation. But some receptors show a spontaneous conversion from non-active to active state in the absence of the agonist, and inverse agonists, unlike full agonists, stabilize the inactive form of the receptor, and that causes to convert receptors to the inactive form, and this decreases the number of activated receptors to below that observed in the absence of drug. Thus, inverse agonists have an intrinsic activity less than 0, reverse the activity of receptors, and exert the opposite pharmacological effect of agonists. So this is a 0, and then the decrease is below 0. So this plot here has an error. 0 should be here. So, and then we go to the antagonists, and antagonists bind to receptor with high affinity but possess 0 intrinsic activity, so thus they don't activate the receptor. An antagonist has no effect in the absence of an agonist, but can decrease the effect of an agonist when present. Antagonists may occur either by blocking the drug's ability to bind to the receptor or by blocking its ability to activate the receptor. So the competitive antagonist, so this is a competitive antagonist, and this is the this is a non-competitive antagonist, this is a competitive antagonist. So competitive antagonists will shift the curve to the right. So it will be identical to the drug. So this is red is the drug, and then the shift to the right will show the drug with the competitive antagonist. With the non-competitive there will be no shift, but there will be decrease in the maximum biologic effect. So these are also the therapeutic index is the difference between the therapeutic dose between the therapeutic dose or therapeutic concentration and the toxic concentration. So the larger is the difference, the more is the therapeutic window. So the wider is the therapeutic window. Like for warfarin, for example, it's very narrow, and for the penicillin it's much more wider. So the wider is the therapeutic index, the more is safety, and the drug is more safe, and that is why these drugs are preferred ones. So thank you for your attention, and that is all for today, and see you next week.